Thank you everyone for joining us for this June 2020 edition of Tennessee Turf Tuesday. I'm Tyler Carr. I'm going to be one of your hosts today. I'm one of Dr. Sorokin's graduate students here at the University of Tennessee. This June edition is titled Getting Ramped Up for Summer Diseases. We have a couple non-University of Tennessee people joining us today. We have Dr. Joe Roberts from Clemson University, Dr. Paul Koch from the University of Wisconsin, and I'm also joined by Dr. Brandon Horvath and Dr. John Sorokin, which you guys probably all know Dr. Horvath and Dr. Sorokin pretty well. Before we get into introductions, I'm going to go over some logistics, some housekeeping items, since we offer pesticide credits for these events. So this webinar will be recorded. It's being recorded currently, and we place these recorded sessions on YouTube. Our YouTube channel is UT Turfgrass, and this will also be available in podcast format. Just on the podcast app, search for Tennessee Turf Tuesday. Pesticide credits are only available for live viewing, though, so keep that in mind. Live viewing only. When you registered for this, Zoom populated all of the information that's required for your pesticide credits. So there's nothing else you need to do, except you need to stay here for the whole entire time. Zoom knows when you arrive and when you leave. And so you need to be here for the entire duration to earn these pesticide credits. Also, if you have questions, we prefer you to feed those through the Q&A box instead of the chat function. So we can answer some of those questions live, since we also have this in a podcast format. And then at the end, I will pull up information for the GCSAA credits, and we'll do that at the end of the hour. So to lead us into our discussion today about summer diseases, Dr. Roberts, do you want to begin with an introduction? Then we can go with Dr. Koch to start this off.
Yeah, sure. My name is Joe Roberts. I'm the turfgrass pathologist at Clemson University. I'm actually based in Florence, South Carolina, which is kind of the central PD region, eastern area of the state. And while we do have a large focus on warm season diseases and nematodes of both cool and warm season, we do have a little bit of cool season turf where we do see some prominent diseases, and a lot of them are highly active right now. So happy to be here for the discussion and look forward to hearing what's going on in everyone else's neck of the woods as well.
Thanks. Dr. Koch?
Yeah, I'm Paul Koch, turfgrass pathologist at the University of Wisconsin-Madison. Been here in various forms for about 15 years or so, and we're just starting to ramp up our disease season. Haven't seen a lot of activity yet, but looking at the forecast, we should be ramping up here pretty soon, so we can talk about
ice just starting to melt off, right, Paul?
Yeah, the ice fishing shacks are in now. Those have been recommended to come in, so we're just getting ready, man.
That's great. Well, thanks guys for joining us. Really appreciate it. I'm Brandon Horvath. I'm the turfgrass pathologist at the University of Tennessee. It's always exciting to be joined by a couple of really good colleagues and friends and visit with you today about turfgrass diseases. And John, you want to go ahead and give a little introduction of you, since you'll probably chime in from time to time?
Yeah, I'm John Sorokin, turfgrass professor at the University of Tennessee, and I'm here just to learn from these three guys a lot. We got a lot going on in our program in terms of trying to establish some cool season grasses during the spring and summer, and so disease pressure is going to be a lot. So I'll be asking questions, I'm sure.
Great. And just kind of to continue a little bit of the introduction stuff, why don't we just go around the horn, Joe and Paul, and I'll share as well. What are you guys working on right now? What's your main thrust of your research program? What are the things that are kind of hot topics for research projects for you? Joe, you want to start?
Yeah, sure. So we obviously we have a lot of warm season turf, and we're about an hour from Myrtle Beach, and a lot of golf courses in the area, and dealing with a lot of root disease issues. And also being in kind of the heart of the Southeast, we also deal with a lot of nematode issues. And one of the things that we've noticed in a lot of our diagnostics over the last few years is we see a really strong relationship with, nematode pressure and some of these prominent root disease issues that we're finding. So that's kind of become a major focal point of our research, is trying to look for links between some of these various nematode populations in association with certain root diseases like take-all root rot, mini ring caused by Rhoxoctonia. And, we see a lot of this because, obviously a lot of these pathogens, be it nematode or fungi, are very prominent. But there are instances where we see differences in control where maybe a nematicide is applied in conjunction with the fungicide. So we're trying to look for ways to mitigate root disease by examining, certain nematode populations in association with areas with high root disease pressure. And then we're looking for ways that can, foster root health. So if we're, thinking about whether it be a disease or a nematode that impacts rooting, if we can do anything to enhance rooting, we're going to be able to, better combat those pests that thrive in the soil. So that's kind of been a major focal point of our research over the last few years.
Cool. And Paul?
Yeah, I would say over the past few years, we've got a lot of different things going on that fall into different baskets, but two of the major areas that we've focused on are sort of, microbiomes and modeling. We've put a lot of effort and resources into investigating the turfgrass microbiome, particularly focused on how it responds to various pesticide programs, and also how we can harness the microbiome for improved disease control. And so I've talked with Joe Roberts a lot on that topic, and we've had some projects that we've worked together on. So that's been one area that we've really focused on. And then disease modeling as well. So, we worked with Damon Smith and Jim Kearns and Brandon yourself on creating the Smith-Kearns dollar spot model. So we still are looking at various uses of the Smith-Kearns dollar spot model, in particular, how to use that model for more precision-based management of dollar spot, and really targeting areas of the golf course that need more applications than other lower pressure areas of the golf course. So really looking at that precision disease management using modeling to help base those applications. And then we're working on a couple of new models for snow molds. So one is for timing of fungicide applications for the traditional, long snow cover duration snow molds like gray snow mold and pink snow mold that we would get here in the Midwest. And then we've also been working for several years with Alec Kowaleski and his team out at Oregon State, and also some partners in Ireland and France on creating a new microdocium patch model, sort of using a similar strategy that we use to build the dollar spot model. So we've been working on both those projects for about 6-ish years or so now, and we're getting ready to sort of create the first version of those models and beta test them in the field. So.
Yeah, that's cool. That's exciting. That's certainly something that's been a long time coming. If you've spent any time up in the great white north with snow cover, and I know, Paul, you've spent some time drilling ice out to measure fungicide dissipation and under snow and ice cover, that those diseases are really significant in terms of the damage that they can cause to turf stands, and having a model to better predict when they're going to occur and get those fungicides out on time so that we get good control is going to be a huge thing for the industry, I think.
Yeah, I mean, I think one thing that we've noticed over the past 10 years is the time where you apply your snow mold fungicides is shifting, and it's shifting year to year, right? Traditionally everybody would have a date on the calendar. You put your snow mold out on that date. The former turf pathologist here at UW, his name was Gail Worf, and he always told our superintendents here in Wisconsin, you put your snow mold out before you go sit in the deer stand. And in Wisconsin, gun deer season is Thanksgiving week. And so you'd always have your snow mold fungicides out before Thanksgiving. But now, we can have some really warm falls. We've had those lately where there's just a lot of rain through early December, and it doesn't make any sense to put out your application at that time. So I think we're trying to get to a more data-driven and weather-based, environmental-based application system compared to just a simple calendar-based method.
That's great.
And Paul, we've seen a lot of variation in warm season fall disease timings as well. Just thinking about, spring dead spot, large patch, for instance, we always like to target, or we tout targeting that 70-degree soil temperature mark. And I've seen such a strong variation in that over the last 5 to 10 years. In my time at the University of Maryland, beginning in 2015, we were beginning those applications in September, whereas a lot of, superintendents that I had worked with, that they had, this idea that even sometimes in August was the right time to apply. And even within the last, 5-7 years, we had, first apps going out in the second week of October. So if you were of that mindset of an August application, and really timing is in October, you're talking about a 2-month window that could be missed. So definitely adhering to those environmental conditions is really important. On the flip side of that, one of the questions that we get often is, trying to target large patch timings. And when you're thinking about managing lots of properties and trying to hit that window, I get questions over, how much leeway is there within that. So again, just reiterating the environmental conditions across both cool and warm season, definitely really important when we're focusing in on these fall, winter diseases. So.
Yeah, for sure. And I think that's a little bit of a segue. I'll just share a little bit about what we're working on. I've got a master's student that is working on a U.S. Golf Association funded project looking at the interaction between the host of dollar spot on golf courses and bent grass, both creeping and colonial, and then the dollar spot pathogen, and then looking at the interaction, the plant pathogen interaction, kind of right at the point where the pathogen infects the plant, and looking at what's being kind of upregulated and downregulated both in the plant and in the fungus to better understand that interaction and provide some clarity to that. And it's one of those things. I'm not that old, but I remember 15, 20 years ago where we used to laugh and wish and hope that we would have a genome sequence of something that was related to turfgrass. And now we have the genome sequences for the host and the genome sequences for the pathogen. And that gives us the ability to do things that, we weren't able to do just, a handful of years ago, which is really kind of exciting to see come to the forefront and be able to be used in something like turf, and better understand what's happening with these plants when they infect the grass plant. You're exactly right, Joe. I mean, that large patch timing has been a question from pretty much day one. I started here in Tennessee. We're in the heart of the transition zone. So Dr. Sorokin likes to teach the students early on that we grow all the grasses equally poorly. And that's pretty much that's from Dr. Powell up at Kentucky, and it's true. I mean, we have right now we have St. Augustine grass at our research farm that has large patch the size of semi trucks, and it's active presently in June. And then, we get into something like zoysia, and then we've got zoysia matrella and zoysia japonica, and we see some differences there. So certainly, being able to understand when those diseases are occurring and when they need to be treated is a key consideration. And I think one of the things that's challenging, especially with large patch, we see it somewhat, and I'd be interested in your comment with spring dead spot too, but we want to be like you guys up north, Paul, and we want to be able to have just, we make 2 apps and then we're good, right? Like for the winter, and we make our 2 apps for large patch, and then we should be covered for the rest of the fall and into the winter and into the spring. And it's so driven by the environmental conditions. We could have a fall that's dry, that is, moderate in temperature, where we might not see a tremendous amount of activity. And then we can have a fall that's warm and wet, and we could just see it blow up, and blow up for weeks and weeks and weeks on end. And that recipe of a couple of applications doesn't really hold. And to some extent, I think that's true with spring dead spot too. Wouldn't you say, Joe?
Yeah, definitely. I mean, I think that with these later initiations, like say we're getting into, the first or second week of October with just starting our applications, one thing that we've seen in ours is maybe pinching that window. Instead of going a 228 day, maybe going down to a 21 day if you're already starting so late, because we know we have an idea of that window of when that fungus is active, or those fungi that cause spring dead spot are active. So, that's one of the recommendations that we have. I know that there has been some instances where there's been some experimentation. I get questions about, spring applications. We actually see spring dead spot develop in December in our neck of the woods. So the damage is already done. So I think, hitting that initial timing and then, taking special care with that window. And, we've even seen some instances where some products at decent rates, have had good control even with a single app. But again, we need to test that in a little bit more detail. So
yeah, for sure. And that's a good segue to, we discussed kind of some questions that we wanted to talk about, and I think this will kind of highlight for the audience some of the challenges that we all face with these diseases, both in the cool and warm season growing regions. But like, if you were going to list, Paul, what would be your top 5, let's say, most difficult to control diseases in the cool season turf region?
Yeah. So, if you go by dollars spent, right? If you're in the upper Midwest, it's probably going to be dollar spot, snow mold, anthracnose, brown patch, and maybe a root disease, right? If you go, southern Midwest, you could probably swap out the snow mold for anthracnose, switch those two around. Dollar spot's not a particularly difficult disease to control if you have the resources and the products available. When we get to, difficulty in control, we're normally talking about something on the roots. And root diseases, just like down south, are so difficult because by the time you see them, the damage is done, right? There's not a whole lot you're going to do to control the pathogen at that point in time. You're really more just managing the stress on the plant. So as far as difficulty, the take-all patch and summer patch, and we're seeing, it seems like more and more Pythium root rot in the Midwest, and it really tends to localize in areas that get heavy spring rainfalls. So we don't see widespread Pythium root rot outbreaks, which is good because we don't have a lot of superintendents getting really devastated by the disease. But it's frustrating because if you're one of those superintendents that happens to be in an area that gets a lot of rainfall for periods of the spring and early summer, you don't have a lot of warning that that disease is gonna come, and it can be pretty damaging when it does come. And just like the other root diseases, once the damage is there, you're playing catch up the rest of the year. So those root diseases are challenging, and Pythium root rot in our region is one that's become more common over the past couple of years that we've been trying to get better information on and get a better handle on when and where it's going to occur.
Right, for sure. And Joe, like, what would you say is the top 5 in the South in the warm season in terms of difficulty to control?
Yeah, I mean, we're still going to be below ground when we're talking about these. And I mean, it's not to say, again, that we don't have foliar disease issues, but, if you're really robust in trying to target root diseases, we see fairly good control of the foliar diseases through those applications. So I would say, take all root rot, mini ring, large patch spring dead spot, and leaf spots, to throw a foliar in there. But Pythium root rot is definitely one that we see a lot as well. It's kind of hard to narrow it down to 5. We see so many.
True.
But we've started to see, Pythium root rot even on warm season. In the past, we kind of, focused more on Pythium root rot for creeping bentgrass, especially, where we are with very intense summer conditions. It's not unheard of, or it's very common for us to see Pythium root rot on bentgrass, grown in our area. But what little bentgrass is still grown here, but we're seeing more Pythium root rot, on Bermuda grass or ultra dwarf greens, and that's definitely a problem. I mean, thankfully we do have some good chemistries that are able to combat that, but that can occur at times of the year, and oftentimes with all these root diseases, you're just really limping these areas along, during periods that it's really tough to get grass growing. So.
Yeah, for sure. And I think that's the common theme, right? Is that when we think about difficulty to control, it's largely underground, the areas of the plant that we can't see very effectively. hard to observe from a a day-to-day basis for a superintendent. They're going to have some form of magnification, whether it's a field scope or something like that. They're going to have to take a portion of the plant out, actually tear it apart and look at it if they wanted to see anything, right? So that's a difficult thing to monitor versus seeing a patch form on the surface. And then by the time that area forms on the surface in the form of symptoms, it's usually long since time to treat down in the root zone. So that makes it especially difficult. And then on top of that, I would argue that I think the other reason why a lot of these root diseases are extremely challenging to control, at least so far, is that all of us being academics and working on problems, and especially if you're a younger faculty member working towards tenure, you don't want to take on a real challenging disease like, take all root rot or something like that. So there's not been a tremendous amount of research done on that compared to something like brown patch or dollar spot or summer patch. And even summer patch, there's been a pretty fair amount of work done for temperature ranges, but we still don't fully understand. I mean, I've had a number of conversations. Paul, you could speak to this a little bit. I mean, there's a lot of folks that think that, bent grass doesn't get summer patch. And I've spoken to a lot of, folks that grow bent grass that have seen some significant populations of summer patch and summer patch symptoms on bent grass in the summer. And then when they engage in a summer patch management program, they see some successful control.
Yeah, I mean, I think that's something that's relatively recent, seeing summer patch develop on bent grass, traditionally thought of as a straight Kentucky bluegrass, annual bluegrass disease. But, that sort of started, Elaine made some of those initial observations years ago back down in North Carolina, and we've seen that spread across, northern parts of the country. We haven't identified any in the Midwest as of yet. Doesn't mean that there hasn't been any summer patch cases on bent grass in the Midwest. It tends to be, higher stress times of the year that we really see that summer patch, those symptoms develop on bent grass. But certainly it can be a very damaging disease. And if it gets misdiagnosed as take-all patch, they have very different control strategies, controlling for take-all patch and controlling for summer patch. So it gets back to knowing exactly what you're up against, so you can implement the proper control strategy. Definitely.
Yeah, for sure. And I think with both with something like summer, because like you said, like traditionally the thought process was always, well, that's take all because it's on bent, right? And there were probably a fair number of those that perhaps were not exactly identified, because you just see the dark hyphae on the roots and say, okay, it's on bent, so therefore it must be, take all, and not necessarily the case. And in terms of the management strategy, you want to just maybe quickly discuss what the differences on those two are? So
well, sure. So the main one, right, is summer patch. You can apply a fungicide during the middle of the summer, right? And you'll see some suppression of control. Whereas with take-all patch, that fungus has gone dormant by the middle of the summer. And when a fungus is dormant, it's not going to absorb, it's not growing, so it's not going to absorb any fungicide from the growing tip of the hyphae. So with take-all patch, we would never recommend a fungicide during the middle of the season because you're not going to impact the fungus. So it'd be strictly sort of trying to limp the plant along until cooler conditions arrive in the fall. Whereas with summer patch, you can apply a fungicide, you can at least arrest further spread of the fungus, and then you can start to implement some strategies to recover some of those, some of the roots that you've lost to infection. So really the use or not use of fungicides is the major difference during the middle of the season for recovery with summer patch versus take-all patch.
For sure. And then there's a little bit of a difference in the window of temperature too, that the pathogen's active, right? So
yep. So take-all patch is kind of between about 55 and 65 degrees Fahrenheit is where it's most active. And in the Midwest, that's normally May, where you're going to make your preventative applications for that particular disease. Summer patch, the fungus starts getting active right around 60 degrees Fahrenheit at a 2-inch depth. And then it's going to continue to increase in activity as the soil temperatures warm. So it doesn't slow down at 65 or 70. It's going to continue to increase in activity. So really, the hotter and wetter it gets, the more disease you're going to see with summer patch, which is not necessarily the case with take-all. Take-all conditions are typically, if you kind of have a mild wet spring followed by a hot, dry summer. Those are prime conditions for take all, and they're not necessarily the same conditions you would see summer patch in. And in the Midwest, we're definitely seeing increasing severity of summer patch, especially on annual bluegrass and Kentucky bluegrass. I would say when I first started at UW, we'd maybe see severe summer patch once every 4 or 5 years, and now it's every couple of years at this point in time. And normally every other year is sort of a pretty severe summer patch year. So warmer and wetter summers are leading to more summer patch.
Yeah, for sure. And then on warm season with take all patch, Joe, you know, I think it's important to recognize that there is a little bit of a difference between the take all that we see on something like bent grass and what we see on the warm season diseases. You want to talk a little bit about those differences, and we can talk a little bit about control strategies and things like that.
Yeah, sure. I mean, in terms of take all root rot on Bermuda grass, I mean, we're targeting those applications starting July 1 is when, you know, you need to be thinking about your take all root rot around that time. And that's where we see that temperature range being much greater than, you know, the Gammanomyces that we associate with bent grass. So, you know, one of the things that we
like 3 or so pathogens that have been kind of indicated that could be causing some of those symptoms.
Yeah, I think NC State identified over like maybe closer to 5. So different Gammanomyces there. I mean, a lot of these belong to the Magnaporthiopsis group, but so they are somewhat related. And I think one of the good things that did come out of the work at NC State was they did show that while there was variation in pathogens, they did have similar sensitivities to fungicides that they had tested. So that was one good thing that in terms of recognizing that. But, you know, we have actually seen very strong differences in start dates for our trials. So like say, we like to look at, you know, having things in place by July 1 to maybe start mid-July. But say we get delayed in terms of trying to get a trial down like we did last year, and we got our first applications down in August. We saw a notable difference in just a 2-week window from trial to trial in terms of activity. So definitely something to think about. And you know, just to reiterate, you know, some of the work that we're seeing, like last year, we decided to throw in, you know, a nematicide treatment with some of our actives in a site where we knew we had a thriving population of root knot nematode. That ended up being one of our best treatments. We actually utilized in the trial, we had a fungicide that didn't actually perform very well in comparison to some of the other fungicides, but when we mixed it with a nematicide to combat that root knot nematode population, it ended up being one of our best treatments for the entire trial. So, you know, definitely, you know, recognizing and sampling regularly to assess where you are in terms of your nematode population and getting a proper diagnosis in terms of, you know, whether or not it's take all root rot or other forms of disease, because, you know, there are instances where Pythium root rot can look like take all root rot from above on Ultrador Bermuda grasses. Just to time in on a very interesting story, we actually put out a take all root rot trial at a local facility more towards the coast that had a history of take all root rot, and ended up being a fantastic trial in terms of getting symptoms to develop. But we noticed that our what we would generally associate as a take all root rot patch, which would, you know, can be about the size of the palm of your hand, we were noticing a little bit larger patches with green in the middle. And we'd pull samples, could find Gammanomyces or Gammanomyces like fungi on there, but we incubated them. Then we noticed that we were actually seeing some hyphae emerging with clamp connections. And going back, we realized that this was actually more of a fairy ring trial than anything. And what we were seeing were smaller fairy rings that developed. And granted, you know, a lot of the fungicides that we were working with were active against fairy rings. But again, another root issue to face. And again, really harping on that proper diagnosis, because Gammanomyces and these Gammanomyces like they're great saprophytes. So they jump on decaying turf, and, you know, being able to associate, you know, symptoms, visual symptoms with signs is really important. And I think a number of diagnostic labs have really moved to that model of making sure that you're submitting pictures with your samples so that we can really get, you know, because again, you know, in some cases, the maintenance plan may be similar. So these fungicides, these dual action fungicides, you know, multi, AIs, they can have broad spectrum effectiveness against different root pathogens. But there are instances where if you have Pythium root rot, completely different fungicides are required for Pythium root rot as opposed to take all root rot.
Yeah, for sure. And could you just speak a little bit to some of the nematode work that you're doing? I had an undergrad that did an independent study for me, and I asked him, what topic did he want to do kind of a deep dive on and dig into, and he said, I'd really like to learn more about nematodes. And I said, all right, well, why don't you go find all the literature you can find on nematodes, come back and give me a list of 8 or 10 nematodes that you think need to be controlled and what their biology is. And he came back and he said, man, everything I can find all these things about all these different nematodes, but it seems like they're really good at identifying these nematodes, but they don't really know what they're doing.
Well, I mean, we're just kind of getting into scratching the surface on, some of our nematode work. But, to start, one of the things that we've done is we've looked at samples across, our region and kind of, stretching up into North Carolina a little bit and just trying to document which nematodes are most damaging. Sting nematode definitely still reigns king. Brent grass or Bermuda grass, as few as 20 per 100 cc's of soil is what we document as threshold. And we see a number of sites where sting is definitely a big problem. And we do work, from year to year looking at nematicides, to try to target timings of whether, we can combat sting. But then also through our samplings, we're seeing, very high populations of root knot nematode. And we have looked at, different extraction methods, much like the work out of University of Florida, where we're looking at mist extraction versus soil extraction. And a lot of our, work has shown, very similar results in terms of in Bermuda grass, mist extraction for endoparasitic stages of root knot nematode is definitely very revealing in certain cases where you can get, very different numbers in terms of a soil extraction versus a mist. And that can be really important for time of year. And that's something that we're trying to look at now is when can we target those nematodes best, because getting to them is going to be best achieved through an ectoparasitic stage. And once they're in the root, they're kind of protected, especially against some of our contact nematicides. So I actually have a student that just started that's going to be working on that and trying to document different soil conditions of whether they may be in endoparasitic versus ectoparasitic stages. And then we also have some projects where we're looking at the timing of when nematodes are a problem versus when the fungi that we're seeing in association with them are a problem. So I have a postdoc that's working on the association between root knot nematode and mini ring, because we do see a very strong relationship between those. So maybe it's a time of year when one is there and one is not. I would imagine they're both there frequently, but very often we see both of those being an issue. And we've actually shown through some of our trial work where we put out a nematicide with fungicides that we know are active against mini ring, we have better control when that nematicide is included and much faster recovery. And again, if you're promoting that root health through different measures, whether it be a nematicide or other means, you're going to get better recovery from some of those fungal diseases. So that's kind of been a focal point of what we've seen. Now, we don't have a lot of work in this area, but we are seeing some rise in Lance nematode populations. And one of the reasons that's really highly problematic is that we really don't have a great nematicide for Lance nematode. So there are instances where Lance nematode can be really detrimental and we see some damage. Oftentimes we're seeing these in mixed populations. So if you can control, sting or root knot nematode, sometimes that can create a niche for Lance to come in and thrive because you're eliminating those other nematodes that may be occupying a space. And that's where we're really trying to look to, just basic root health measures. So can we employ certain, compost, or we're looking at biochars, anything that we can do that may not necessarily impact the actual nematode population. And that's not to say that those products may not do that. There's been some great work out of the University of Florida where they've shown certain compost to actually reduce sting nematode population. But because we see a very direct link with plant health and nematode damage, if we can employ some of the benefits of certain amendments that may enhance nutrient retention, enhance drought tolerance, that's going to just help those plants to better survive amidst those high populations of nematodes.
Yeah, for sure. Because besides just the association with the pathogens, I mean, I would suggest that there's probably a reasonable hypothesis that you could make that, any of those areas on putting surfaces, whether it's on warm season turf or even cool season turf, right? That those areas that you constantly, that this area seems to just, dry out and look a little rougher in that spot every summer, all the time, that there's probably something more than just the fact that that's in a little bit of a high spot and it just dries out a little bit more, but there's probably something munching on it and, underground, and there's a potential for those hot spots or areas that just seem to always be problematic, that there might be something biological there. In terms of sampling, if you were going to recommend to a superintendent what a sampling protocol would be, what would you say as a way to kind of get started understanding what your nematode impact or nematode load is in your system?
Well, I mean, first and foremost is if you do have some of those defined areas, like you're saying, that are traditionally weak, sending in a diagnostic sample, would be great to just get an idea of what numbers and where you stand. I mean, a lot of times we see these areas pop up in transition periods. So like now, where we've had a really awkward spring, we've been very dry. A lot of guys are relying more on irrigation water. So if you have poor irrigation sources, you're stressing out, the turf in that manner. And if you're not getting that general response that you normally expect with a transition, again, whether it be cool or warm season, and you've ruled out other factors. So say, I don't really think I have compaction, within those areas. That might be a sign to also, look for a nematode sampling. So differences between routine sampling and diagnostic sampling would be, if you have a defined area of declining turf, that would require more of a diagnostic sampling. And much like we talk about in diseases, we always want to take from that more outer region, because that's where those nematodes are thriving. So nematodes need a living host. They don't want to feed on dead turf. And if you've got an area of dead grass, they're going to be, moving outward from that area to look for more healthy grass. And oftentimes there are instances where you may find higher nematode populations on healthier grass because that root population can support a higher, threshold of nematodes. So, we would just recommend, pulling, cores from an outside area of a defined area declining, or if you're thinking about more of a routine sampling, break up your greens and, just do more of a systematic sampling across a given area, 15, 20 plugs, half-inch soil plugs, and then, send those into a diagnostic lab that could identify, nematodes at your particular site. Those are kind of the differences between that. Now, if you do have a history of root knot nematode, and especially if you're managing Bermuda grass, that's when you're getting into the idea of sampling utilizing the mist extraction versus the soil. For bent grass, there's really not much difference, and I think that was really shown, through the work at University of Florida. But if you're managing Bermuda grass and you have a known history of root knot nematode, looking at, a mist extraction could be beneficial in determining how many juveniles are actually in the roots, and there are, variations and thresholds for that as well. So,
gotcha. And then Paul, are they the folks up in the Midwest talking about nematode impacts and things like that?
Yeah, definitely. It's something that we're talking about more than we did a few years back. One of the problems though is we don't have a real good idea what the thresholds are up here, and so you can get some samples that have pretty high counts, but like Joe was just talking about, healthier turf can oftentimes support higher populations of nematodes.
Yeah, aside from Sting, I don't think we have an idea either.
Yeah. So my take on nematodes in the Midwest is that they're stress. They are one of multiple stresses that can happen. We tend to see nematodes in the cleanup passes and the walk on, the walk off areas, just like we tend to see other root impacting diseases. When Joe was talking about was talking about the sort of the overall root health issues on nematode issues. It was making me think of a take-all trial that we did a few years back on a fairway up in northeastern Wisconsin, and the superintendent had he wanted to keep the carts from going on our trial, so we had the cart signs directing traffic, 3 feet in front of our take-all trial. The take-all on the other side of those cart signs was incredible. It was awesome take-all. We had no take-all in our trial at all. Now, you can't tell me that there was a whole load of fungus 3 feet away on the other side of the cart signs, and there was no fungus in our trial. It's just a matter of the plants were healthier. They were less impacted by traffic. And so they had a healthier root system. They were better able to fend off against the amount of fungus that was present there. And I think we see the same thing with nematodes. But certainly there's been cases where nematicides are needed. And we just need more information on what sort of thresholds exist for our courses in the Midwest, and have a better sort of strategy and plan for when nematicides are best used for nematode control up here.
Yeah, for sure. Speaking of that, that's so funny that you mentioned the cart traffic and the movement of the fungus away from your trial. We were, Joe mentioned mini ring is another one that we have challenges with in terms of disease timing, and one of the things that I think is very difficult for the warm season folks with Bermuda grass is that on bent grass, you're moving from when the diseases are active towards a period of recovery, right? Like diseases are generally active spring, summer. Yes, there are fall diseases, but you're moving towards that window of recovery for most of the big diseases. Snow mold would be an exception where you got to deal with it, and you might recover a little bit before you get into summer, but that's maybe one of those more challenging ones. But for Bermuda grass, one of the things that I think makes some of these diseases extra challenging to know and to be comfortable controlling is that these things are active when you don't see symptoms at all, like because the grass is so actively growing, right? Like mini ring, the pathogen's going to be active. It's Rhizoctonia zea. It's the same Rhizoctonia that we see leaf and sheath blight on bent grass in the hot, really hot summer. That pathogen's active in the middle of the summer on Bermuda grass as well. You don't see any symptoms at all. And then in the fall, when things start to slow down, you start to see damage and you go, oh, I have a problem. And you were talking about that cart thing. I'm just going to share my screen really quick and show this mini ring trial that we had, because it's a funny, add on to that story. But this is the non-treated control. So as you know, when you go off site, you call superintendent and they go, oh, I'm loaded with that disease, whatever it is, right? And that generally means they get like 2 patches, and it's not going to be where you put the trial. And so we put this trial out on this local golf course on Ultradwarf, Bermuda, and we started apps in mid-July. Joe, you were talking about take all and mini rings, kind of the same thing, mid to late July for application timings. And we put this app out and we go, two full cycles of apps, no disease apparent at all. So I'm worried about getting data. So we go to another place that's starting to see some symptoms. We put the same trial out there. We get no control out of any fungicides because we applied that at a curative timing, and we just weren't seeing any recovery from those symptoms. About another week goes by and this trial just blows up with mini ring all over the place. So there's non-treated, there's Calida, Navicon, another untreated, and then another Calida. And it was pretty funny how, how we saw this, disease pop up. And just, it was amazing to me that, the pathogen was present. It was active. We were making applications that were controlling it. And we didn't see that until we got to the period where the grass started to slow down and you saw symptoms develop. And similar to your point, Paul, with the moving the traffic, you didn't move the fungus, right? You changed where the stress was occurring. And it goes right back to that basic concept of disease triangle. If you have a more susceptible host, that's where you're going to see some disease activity, right?
Guys, let me chime in here. We have a couple questions in Q&A.
Yep, I just pulled them up too. Go ahead.
So the first one is, do you have any visual examples of what nematode damage looks like?
I think Joe can probably throw up one or two.
And while Joe's doing that, how important, Joe, are the use of wetting agents to help if nematicides are contact to use when you're for treatment?
Yeah, so so here is a very characteristic area of sting damage on Bermuda grass. I mean, notice it's somewhat localized, but very diffuse in terms of modeled where it is. And basically, I'm not sure if I can figure out how to do the pointer, but you would just want to pull from.
You can see your mouse moving.
Oh, okay, you would just pull from that that outer region. You definitely don't want to pull from these dead areas, because the nematodes are definitely going to move. But in this case, yeah, we know that sting was here because this was we actually did sample. And one other telltale here is just looking at those roots. You can see these short and stubby roots, and this is really characteristic of sting nematode damage. Now, in terms of nematicides, for sting. We've got a couple of different nematicides that are very effective. Indemnify is highly effective. We also abamectin or Divanem, which is another trade product. With abamectin, for instance, being a contact nematicide, we definitely recommend a wetting agent in the tank with that particular nematicide, because you want to be able to penetrate that down into the area where those nematodes are active. So definitely following label recommendations. But one other thing that we also recommend with abamectin is the use of estrobulirin, and I've seen some very good results in root health and overall improvements from that combination as well. So again, that those combinations are definitely really important. And we see areas of LDS, and those can have lower levels of nematode damage, and again, that's where another wetting agent would be really important. So
yeah, for sure. And I think the other thing too, Joe, and I know you've seen this as well, is that with fungicide applications as well, especially now as we're recommending more and more fungicide applications get watered in for a systemic activity, that having that wetting agent program in place before you're making those applications is a key thing too, because it does take some time for those wetting agents to get active and start making that wetting front in the soil more uniform and those kinds of things. And in order to do that, you've got to have that wetting agent program already down and in place in order to make sure that whatever you're going to apply and then water in is uniformly distributed in the soil. So that's a key consideration, both from nematode applications as well as fungicide applications.
Yeah, and I've gotten a question about that recently, thinking about wetting agent programs. And we, just like you said, Brandon, we definitely recommend being on a wetting agent program. But sometimes I'll get a question of, well, I mean, I've already put out my wetting agent. Am I going to hurt my greens by applying too much wetting agent? And so all of our nematicide trials that we've done, and typically, for specifically for abamectin, we're always applying that with a wetting agent. We're applying these on areas where there's already an established wetting agent program. So, we may be utilizing, something like Fleet or Revolution on a regular basis. So say like a monthly application of 4 ounces, in the case of some of our maintenance applications. But even on top of that, for an abamectin application, we're going out with a wetting agent included in that tank mix as well.
Yeah, absolutely. And I think the key there is just making sure that there's some space between those applications, right? Like the only time I would be concerned of overloading the system with a wetting agent would be if you were, going back to back with applications and really, greatly increasing the potential rate that you were putting out. But for the most part, that's not going to be an issue with those, maintenance applications versus a targeted app like a Nematode app.
Exactly. And you could also time your wetting agent application with your nematicide. So if you're on a schedule for your wetting agent program and you have concerns over applying too much wetting agent, try to time your application so that you're getting that nematicide down when your wetting agent is needed. That's also another way to look at that in terms of product use and efficiency.
So as the non-pathologist, would you want to separate those applications with your nematicide with the wetting agent to get it into the soil to where the nematodes are? But if you're going to follow it with or use a strobe as well, would you want to put that as a separate application so you can get the systemic uptake by the plant, so you get that?
Yeah, so we're
tank down in the soil as well.
We're tank mixing all those together, so we don't see any detriment to putting a strobe in the same tank with the wetting agent with the nematicide in the case of that. So it depends on what you're trying to target. If you're really trying to drive that product down and get that uptake in the roots, we don't see any detriment to putting all three of those ingredients in the tank.
They can be taken up by the roots, but I just wasn't sure if the roots are compromised, would you want to, if it's got a bicipital or a bicipital movement down to the roots, you want to get it. You want that strobe to protect the plant by getting it on the plant if the roots are being compromised.
Yeah, but in terms of that application, we're really trying to drive it down into the roots. So, we wanted that product there, but if we were looking at more of a plant or upper protection, maybe it would be making more sense to have it as a separate application. But because we're seeing the benefit of this with the combination of nematodes and potential fungi that are existing in that system, that's where we're seeing the benefit of the three-way tank mix.
Yeah, for sure. Another question that came in is about residential lawns and active areas of research for disease control in residential lawns, and what are good sources of information. I would say, and you guys chime in, the big areas of research for residential lawn in terms of disease control, certainly in our area is on warm season grasses. It's going to be large patch predominantly, and then maybe a little bit of spring dead spot, but mostly large patch on zoysia grass. And then in other residential lawns, it'd be brown patch and tall fescue is probably the big one that folks need to control in this area. And then cool season wise, I would suspect that Paul, it's probably necrotic ring spot, summer patch, that type of stuff on Kentucky bluegrass or mixtures of Kentucky bluegrass.
Yeah, so necrotic ring spot, summer patch on Kentucky bluegrass. We have research going into various diseases, summer patch, red thread, snow molds on fine fescues, for that particular species. But the vast majority of disease problems on lawns in the Midwest are necrotic ring spot and summer patch on bluegrass.
Yeah, for sure. And then in terms of good sources for information, I mean, I would say that your extension pubs in your various states are probably a pretty solid source of information. And then in terms of fungicide control, staying on top of what's being put out at NC State and the turf files, there's a pretty good resource of information. We're getting ready to add diseases to our mobile weeds app, and it's going to become the mobile pest app. So that's that will be an area of information. We're loading some of that efficacy information in there now. And then certainly the document that you contribute to, Paul, and you could speak to this, is the PPA one that's done with you, Paul Vincelli and Bruce Clark, right?
Yep. So I've been involved with that publication the past couple of years as Paul Vincelli is sort of gradually moving outside of turf. We are working on a new update. We're not going to have it out probably this year, but we're just making some final updates to that and either have it out later this summer or over the winter. And we also have similar to what Brandon talked about, we also have a mobile online version of that we host at the University of Wisconsin, which is turfpests.wisc.edu. So it's basically just an online searchable format of that Kentucky document.
So Brandon and guys, we have more and more in East Tennessee, especially the upper East Tennessee, we have more and more bluegrass lawns going in. So with brown patch, Rhizoctonia being the big thing on the tall fescue lawns, and summer patch, obviously on the bluegrass. Control for those for home lawns. Is it the same products? Is it the good strobies again? Like
Yeah, they're gonna be different. Strobies are gonna be solid on both of them. The other thing that I would strongly support would be, just like, up in the Midwest, the three-way mixture of grass seed, of fine fescue, perennial rye, and Kentucky bluegrass is usually a pretty solid setup for a home lawn. In our area, I would highly recommend neither a mono stand of tall fescue nor a mono stand of Kentucky bluegrass, but a very strong mixture of the two. I did research when I was at Virginia Tech where we looked at mixtures, and when you looked at a mixture of 50-50, close to a 50-50 on seed count, right? So that's going to be a lot more Kentucky bluegrass seeds than tall fescue seeds in terms of percentage, if you look at the overall amount, but there's a lot more of the seed, a 50-50 mixture on seed count. You saw a reduction in both summer patch and a reduction in brown patch because you had enough non-host in the way that you weren't able to really get a whole lot of disease activity going on either one of those. And so I think that's something that we'll want to consider as we start to see some of these heat tolerant bluegrasses that look really good kind of move into our area for sure. Another question, and Joe, you can speak to this from a diagnostic perspective. Is there a website or email address we can send pictures to help identify diseases? This is the hardest area for me to identify. And all I would say is we don't have a diagnostic clinic that I run. Our state diagnostic clinic over in Nashville does take samples. Pictures are, if I send you a picture like this, that's pretty easy to figure out what that is if I tell you it's Kentucky bluegrass in the summer in Cincinnati. But at the same time, I think, I always like to say that textbook pictures are textbook pictures for a reason. And very often the pictures that you have of something happening on your turf stand isn't necessarily a textbook picture. So you want to speak a little bit about the diagnostic process and the challenges associated with picking out a disease on pictures?
Yeah, sure. I mean, really, in terms of, if you're having difficulty with diagnosis, utilize your local diagnostic office or clinic, should you have them. And, as far as resources, there are plenty of resources where you can find pictures and you can find variation in textbook pictures. But, we pick those and get them on the web because they're very good visuals for, seeing these. But just like I had mentioned earlier with the fairy ring take all root rot, conundrum that we were seeing, it really takes a trained diagnostic eye to be able to differentiate some of these. And, a lot of these fungi that we work with, they're incredible saprophytes where they, may not be the necessarily the pathogen in that case. And sometimes there are similarities in the way that these look. We have issues where we see a lot of curvularia that can come in, or leptospirulina, or, these are things that come in and it may look to an untrained eye that these are the causal agents, but they're really just great saprophytes jumping on dead turf. So definitely recommend, utilizing a diagnostic lab for being able to differentiate, symptoms. So something like, I know Brandon's showing some necrotic ring spot, but, thinking about, tall fescue, gray leaf spot, Pythium blight, and even brown patch in certain cases, those can have similarities in their symptoms. And it just takes years and years of experience and high powered microscopes to really be able to differentiate these in a lot of cases. There are, some instances where a picture can be very, helpful, but it's not the end all in terms of being able to form an accurate diagnosis. And that's really critical when trying to address control issues. So.
For sure. Well, I think we're out of time. Tyler, you want to handle the particulars for the GCSAA education points?
Yeah. So for GCSAA education points, this is your event approval code. And you need to make sure that you put down this event date, whether that's being, whether you're watching this live or as a recorded version. This is the information you need in order to obtain these pesticide credits. So
Great. And then Tyler, there's a question in the Q&A about just passing pesticide application certification exam on June 1st, still waiting for license. Daryl Hensley told me to reach out to the chair. So for the person that asked this question, we'll go ahead and get your name and information to I would say Dr. Brosnan, who is kind of the organizer of our Turf Tuesdays, and get him that information so that he can follow up with you to resolve that issue accordingly.
yes, I'll type in his email address, Brosnan.
Yeah, let's do that. That'd be good.
It's Brosnan at utk.edu, right? I believe
that's right.
Okay.
Since you just gave that out to everybody.
I mean, it's on the website. It's not like he's young.
Yeah, no, I know. It's Jay Brown.
I'm going to stop the recording.
And thanks everybody for being here. Just thank you to all the attendees. Hopefully you got something out of this, and really appreciate y'all being here and paying attention. Joe and Paul, thanks for being willing to come on and chat about diseases. Always exciting to see you both, and look forward to seeing you in person somewhere soon down the road.
All right.
Yeah. Thank you guys.
Thanks, everybody.
Thank you.